This lecture is about how populations evolve. So we'll start with a brief description of the role of Charles Darwin. When most people are asked to give their knee-jerk response to what is evolution or tell me something about evolution, the first name that comes to mind is Charles Darwin. Now, I don't want to discount the fact that his work was absolutely pivotal to the understanding of modern evolutionary biology, in that Charles Darwin proposed the first correct mechanism for evolution, which was natural selection. There were other people who were his contemporaries and people who lived earlier than Darwin who did also come up with ideas for how evolution worked, but their mechanisms were actually not correct. One of them that you might have heard of is Lamarck. He came up with this idea of acquired characteristics. So, for example, if you are a browsing animal and you stretch your neck really, really tall to eat off the trees, your offspring will also have longer necks, and so on and so forth. But that actually is not a correct mechanism. However, natural selection is. And we'll get into more detail about that. But I do want to stress to you that over the last 150 years, Darwin's ideas of natural selection have actually been melded and synthesized with many scientific fields that are strongly quantitative or measurable. So we're talking about genetics and statistics, paleontology, botany, geology, ecology, biogeography. And now we refer to it as the modern evolutionary synthesis. And this is a more refined and full concept of how evolution works. If you'd like to learn a little bit more about it, feel free to click on the little link, the Wikipedia link to modern evolutionary synthesis. But as this course goes on, you'll learn a little bit more about this. So, we need to distinguish between something called macroevolution and microevolution. Macroevolution is where you're looking at all of the diverse organisms on the planet. So the evolution of new species, new groups of organisms, dogs, cats, trees, birds, insects. How did all of these things arrive? Well, we have many lines of evidence for macroevolution being an important force of evolution, and we'll go into those in a small amount of detail. But most of this lecture will actually be focusing on microevolution, and that's the evolution that happens within a population. First, let's have a look at lines of evidence for macroevolution. These are similarities among living organisms and distributions of living and fossil organisms in time and space. There is a huge amount of research and many great resources for macroevolutionary evidence, and I've given some links below. But let's look at a few brief ones. The first is similarities amongst living organisms at the cellular level. We actually have something called cell theory, and remember, when I say theory in biology, I'm talking about something that is a broadly correct fact, not talking about a hypothesis or something that hasn't yet been proven. We're talking about something that there's a lot of evidence for. So, modern cell theory. All known living things are made up of one or more cells. All cells come from pre-existing cells. The cell is the fundamental unit of all living organisms. All cells have got DNA, which codes for proteins, and all cells have basically the same chemical composition. So we're talking about phospholipid bilayers in their membranes. We're talking about proteins having similar functions. We're talking about the energetics of cells having similar functions like ATP and respiration. You can compare Golgi apparatus in an ant and Golgi apparatus in a human being. And there are very, very strong similarities across all organisms. At the molecular level, because all organisms use nucleic acids, DNA and RNA and proteins are really information molecules because they retain a record of the evolutionary history. So imagine this. Every time that a cell replicates and a mutation occurs in the DNA, that mutation will be recorded in the DNA. And if that organism's managed to survive and have offspring, that mutation will be copied down from one generation to the next. So you can actually compare nucleic acid sequences, protein sequences between different organisms to estimate how similar or how different they are, and we use that to reconstruct evolutionary relationships. So the more closely related you are to another organism, the fewer differences you'll have, and the more distantly related you are, the more differences you'll have. And this has been really useful in figuring out relationships between things. For example, in the old days, we used to talk about mammals, birds, and reptiles as being 3 separate groups. Well, we have now got very good data from both fossils and molecular evidence that birds are actually evolved from a lineage of dinosaurs that were the sister group to crocodiles, and birds are actually within the reptile group. So birds are warm-blooded reptiles. So that totally changes how we think about the relationships between organisms. Developmental biology is really useful. So the example I have here is about whales. Now, whales evolved from an ancestor that was a hoofed mammal, closely related to hippos. And there's a great animation that you can watch. The link is at the bottom here. It's of the evolution of whales, and it shows you how over time you had something that looked a lot like any other hoofed mammal, slowly evolving to become more aquatic. Now, the ancestral hoofed mammal had teeth. There are whales that have full sets of teeth throughout their lives. Having teeth is an ancestral state for whales. There is a group of whales called baleen whales, and they don't have teeth when they're adults. They have a filter feeder instead inside of their mouth, and that allows them to filter out small creatures. But baby baleen whales, while they're still a fetus, actually have teeth in their jaws, and then they lose them before birth. So that gives you some evidence for common ancestry with toothed whales and other mammals. Anatomical structures. So homology is the existence of a shared ancestry. Now, if you look at the bones in the human, dog, bird, whale, you can actually see the homologous structures being represented. Now, if you're a little bit dubious about this, there's actually some extraordinary research done with fishes that indicate that a series of genes called Hox genes, which are actually found in fish too, they're involved in the making of fish fins, are involved in the making of human, dog, bird, whale limbs. And it's as simple as this. It's a bit like an orchestra playing a complex piece of classical music. For each musician, the musical score tells the musician when to begin playing, how fast to play, and when to stop playing. And in the human, the dog, the bird, and the whale, they each have this composition, but the difference is how fast and when each of the players is playing. So the genes are the same genes, but what is different is the regulation of those genes, when those genes are being switched on and when they're being switched off. So for example, the gene that's involved in your radius, that's one of the bones in your arms, that gene is switched on and creates a much longer radius in you than in the whale. So we have homology both on the structural level and then also on the genetic level. Now, selection is something that happens in nature, so natural selection, and that's in response to natural environmental pressures. But humans have been selecting organisms for millennia, and we call this artificial selection. And artificial selection is useful because it can give us an idea of the mechanisms of natural selection as well. So for example, wild mustard, which you can still find in some places, has been selected for over millennia in different ways, and as a result we've got all of these different brassica vegetables like cauliflower and cabbage and kohlrabi. But the thing is these are actually still just one species. You can actually breed them with each other and create hybrids between kale and broccoli or cauliflower and cabbage. And the large majority of human crops have actually undergone such strong artificial selection that you would be challenged to find the wild relative of the commercial crop. A well-known one is teosinte, which is this tiny little corn-like creature on the left-hand side. And if you compare that with modern corn, which is absolutely enormous in comparison. This link at the bottom here for crop wild relatives goes over a variety of different human food crops, and it's quite interesting if you are into that. Okay, so now let's start transitioning into thinking about populations as units of evolution and microevolution. So just to remind you, a population is a group of individuals of the same species living in the same area at the same time. And this is actually the smallest biological unit that it can evolve. Individuals cannot evolve. I can't evolve. I either live or I die. But over time, our population can evolve. So let's get into that in a little bit more detail. So a gene pool is a description of all of the alleles of all of the genes in all of the individuals that make up a population. And microevolution is the definition of the change in the frequencies of these alleles and genotypes. Evolution is a quantitative science. Now, if I told you civil engineering is a quantitative science, you wouldn't be particularly concerned because you would have an idea that in civil engineering you're going to be measuring the distances of roads or the widths of roads or how hot they get or the strength of a bridge. Well, microevolution is a quantitative science. We actually can measure the amount of evolution going on in a population. So how do we do that? Well, let me show you. We would need to calculate allele frequencies and genotype frequencies, and we would need to do that over time. It's a bit like my weight. If I measure myself just once, I don't know if I'm getting bigger or getting smaller. So I can't say anything about my weight gain. I can just give you a single point. In order to give you an idea if there was some change going on, and evolution really is change, you would need to measure my weight at separate intervals. Now, would you do it at 12 o'clock and 12:01 and then say that I'm gaining weight? Probably not. You need to have a reasonable interval of time. So for most organisms, you would want to think about what's the breeding interval at which you've got births and deaths occurring. So for some organisms, that might be over a period of weeks, others it might be months, others it might be decades. It has to be appropriate to the organism. So allele frequencies are the frequencies of your recessive allele to your dominant allele. Your genotype frequencies, on the other hand, are the frequencies of the homozygous dominant, heterozygous, and homozygous recessive genotypes. And we can actually go into a population and count those things. So to figure out the frequency of your recessive allele, what you would do is you would count the number of copies of your recessive allele. So if you go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 little b's over 26 total number of alleles, that's a frequency of 0.58, or 58% of that population is little b. And then you can do the same thing for the frequency of your dominant allele. I want you to notice how the frequencies add up to 1. In order to figure out your genotype frequency, you basically do a similar thing. You would count how many incidences do you have of homozygous dominant. So for example, you've got 1, 2, 3, 4 homozygous dominant individuals out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So it's 4 out of 13 is your frequency of your homozygous dominant. So you can go into your population, you can measure your allele frequency and your genotype frequency on multiple different occasions, and you look to see, are they different? If they're exactly the same, you would say that there's no change, no evolution. And if they are different, you would say there is change, so evolution has occurred. So then the question is, well, what kind of evolution? It turns out that Darwin came up with one of the causes of microevolution, but there's actually 4 of them. There's genetic drift, gene flow, mutation, and natural selection. So in genetic drift, you basically have a small population that's so small that just due to chance, you may lose individuals. Think of it this way. If in your family, one person in your family had blue eyes and everybody else had brown eyes, they've got the gene for blue eyes, but it could just be the luck of the draw when they had children that all of their children ended up with brown eyes, not blue eyes. And so that blue-eyed allele would disappear out of the population. And in this picture that you see below of the red and white flowers, that's pretty much what's been happening in the flowers. So it's only the flowers that were red that happened to leave offspring. And so over a number of generations, the white flowers disappeared. It wasn't that they were not well suited to their environment. It was just the luck of the Punnett square of which offspring were created by the crosses. There are actually 2 subcategories of genetic drift that are interesting and you should know about. One of them is called the bottleneck effect. In the bottleneck effect, you have an original population that has been reduced dramatically. And when you reduce the size of a population, you're losing the genetic diversity within that population. And so your surviving population may not represent all of the original variation. And this is extremely important for endangered species. For example, the cheetah, the Florida panther, the prairie quail. All of these organisms suffered from genetic drift when the variation in their alleles was lost. And you might not think that's a big deal, but when the surviving population doesn't have a lot of variation in it, it creates a situation which is very similar to if you were forced to breed with your brothers and sisters. And you know from humans it's not a good idea to breed with close relatives because you're going to end up with a lot of your recessive alleles, your disease alleles, popping up, and that actually results in some genetic problems. And this is happening to many wild creatures whose populations are becoming too small. Another version of genetic drift is called the founder effect. And this is when you have a small number of individuals from a main population that move to a new location. But that small number of individuals is only representing a small proportion of the total genetic variation. And if those founding individuals in that new population then breed, they're breeding with close relatives, and over time you can have disorders arising within that population. A classic example in humans is Ellis van Creveld, and it's found in the Amish and in some small islands. It's very rare, but it's found with much higher incidence in these founder populations because there's less variability. And if you're interested, you can always read more. Follow the links. Okay, so the second is gene flow. This is the change in a gene pool by individuals coming into a population or leaving the population. And the example that I've given here is of red hair. So if all redhead people were to leave Scotland, the next generation there would likely have very few people with the trait. And we would say that the Scottish population has evolved. Another example of gene flow is when you have people going to other parts of the world and having children. So an example is US soldiers in Vietnam. If they had children with women in Vietnam, they're adding their European alleles and genes to that population there and changing the gene pool frequencies in the Vietnamese population. So we would say that evolution has occurred. Mutation. So we've talked about mutations a couple of times. Errors in DNA replication can lead to mutations. Some of these can be lethal and some of them can be advantageous. So the example that you have here is of a mutation that resulted in a different colored pigment in a plant. This is a mutation that could be passed on to the next generation, and if for some reason having orange flowers was of more advantage than having yellow flowers, over time natural selection would select for your orange flowers. And then of course natural selection. And of all of the causes of microevolution, only natural selection results in adaptation. The effects of all of the other forces are random in that they're sometimes beneficial and sometimes not. And here's an example for you. Before selection, if you have some bacteria in your body, there's going to be variation in those individual bacteria. You take some antibiotics and it kills most of your bacteria, but some of them, because of minute differences in the DNA, may be able to resist being killed by your antibiotic, and then those ones will breed and your final population will have high resistance to antibiotics. This is happening in humans, it's happening in our food supply, and it turns out that we're actually probably running out of antibiotic that will work for most of our disorders and diseases. So for example, there is gonorrhea that is now antibiotic resistant, and chlamydia that's antibiotic resistant. And many of you have heard of MRSA or methicillin resistance staphylococcus infections. So it's something to really be aware of.